I'll start it. Well, let's see what we can do. Um, so today we'll be talking about this, and of course Horst had drawn all these nice pictures, which is why I didn't really want to delete his whole presentation. He's very much into pictures. But the point of this slide was that CO2 is a byproduct of all the metabolism that happens in the mitochondria. CO2 rapidly diffuses through membranes, being neutral in electrical neutrality, and so there's this large amount of CO2 that gets produced, and it's like a couple hundred liters per day of CO2 that has to be eliminated through the lungs, and it gets transported from all these distal tissue cells, mainly through blood cells. And that's because CO2 diffuses into the blood cells, and then carbonic anhydrase converts it into bicarb and hydrogen ions. And the hydrogen ions can adhere to the hemoglobin, especially in a deoxygenated state. Hemoglobin is a very good hydrogen proton acceptor, and anionic areas of the hemoglobin are exposed. It binds the hydrogen ion, and then the bicarb sort of gets trapped in the red blood cell, after this all occurs, and so it all kind of gets transported back to the lungs. In the lungs, sort of the opposite thing occurs. Hemoglobin takes on oxygen. The conformation change helps the hemoglobin dump the hydrogen ion, and the bicarb combines with the hydrogen ion to produce CO2 that diffuses back across the cells, and the CO2 is blown off through the lungs. In addition, to maintain all the electrical neutrality that has to be maintained, there's a lot of cellular pumps going on, and a lot of exchange pumps. So anions get exchanged, and chloride and bicarb get exchanged throughout the system, both in this system on the cellular membrane, but in the kidneys as well, where renal excretion of bicarb occurs. It's kind of tied to chloride. So, see, I think that was all in the same thing. So we have, in addition, and proteins with their anions make are a major buffer internally in the cells, intracellularly, I should say, as well as phosphates. And the bicarbonate buffer is sort of the extracellular, mainly the extracellular buffer, along with the addition of ammonium. So, kidney function, in the proximal tubules, of course, everything is sort of filtered through the glomerulus, and in the proximal convoluted tubule, you're absorbing water and sodium and all these things. You're also reabsorbing bicarb, mostly in this area. And then in the loop of Henle, there's the countercurrent mechanism for absorbing even more sodium and chloride it back out of the tubule and into the blood circulation. And then in the distal tubule, there's an opportunity to actually secrete acids. And so in the distal tubule and in the collecting ducts, you're secreting potassium, acid, and ammonia, which is sort of a weak acid that the kidneys use to kind of do acid-base balance as well. So bicarb and sort of these weak acids are the main mechanisms for the kidney to compensate with acid-base metabolism. Horst was making the point here that really whenever we talk about pH, we're talking about the concentration of hydrogen ions, and the concentration of hydrogen ions is actually very small in comparison to water, the rest of the cellular system, and the dissociation, let's see. Oh, well, I didn't know it did that. Okay. So the concentration of hydrogen ions is really, like 0.00000. We're talking about nanomoles per liter. And they take the negative log of that concentration in order to get to 7. So, a pH of 7, tells you have like 7 decimal points. The pH of 7.4 is basically a nanomolar concentration of 40 hydrogen ions per liter. So that's actually very few hydrogen ions in comparison to the other things. And whenever we talk about other electrolytes, we're normally talking about millimoles per liter, not nanomoles per liter. So this is something that exists, in sort of another thousandth as small as in comparison to most of the other electrolytes that we're talking about. But nonetheless, it's very tightly regulated, and a change, for example, from a pH of 7.4 to a pH of 7.6 doesn't sound that big, but that's decreasing your hydrogen ion concentration by almost half. So it's actually pretty dramatic, and that's how strong the hydrogen ion is. Similarly, a 50% increase in your hydrogen ions will give you a pH in a 7.1, 7.2 area. So if you think of it in that terms, you can see why these are actually large swings in a relatively small amount of hydrogen ions that are tightly regulated by the body. And whenever you start getting outside of that 7.2 to 7.6 range, you're pretty deathly ill. That's a pretty big problem for the body, and it does tightly regulate that. So there are several different ways to think about acid-base balance and to calculate it and all that sort of stuff. Sort of the older approach is what's labeled here as the physiologic approach, and that's really just kind of concerning yourselves with the amount of bicarb buffer in the system relative to the amount of CO2 acid in the system. Let's see. So, oh boy. Okay. So mainly what I wanted to show here was that this system relies on sort of just the balance of the bicarb with the CO2 to try to keep you on this line, on this graph, with a pH of 7.4. And so we're not going to do all these calculations. I'm not calculation heavy. As we can see, Horst is a mathematician, not myself. So he can explain much of that. But basically, in this paradigm, you're sort of balancing the CO2 with bicarb. And the reason why he's drawn the bicarb very large is because bicarb is not really that strong of a buffer, and it takes a lot of bicarb to sort of offset a little bit of CO2. And I think the ratio is something like 20 to 1 with the sort of volume amount needed. So if you have extra CO2 in the system, then in this system you can counterbalance it by just adding some more, by the kidneys not excreting as much bicarb, holding on to a little bit more bicarb, and trying to sort of rebalance things. So from the kidneys' perspective, that's mainly what they're trying to do. So you can, in this system, you can have a normal situation. You can have acidosis and alkalosis, but if you have acidosis or alkalosis, then if you have acidosis, the kidneys are going to hold on to more CO2 to compensate. If you have too much CO2 from compensated alkalosis. So this doesn't look right to me. All right, so if you have too much CO2, if you're retaining too much CO2, then you can retain. If you have too much bicarb from a metabolic alkalosis, you can also compensate respiratorily by holding on to more CO2 or not blowing off as much CO2. And that all comes from the Henderson-Hasselbalch equation. Graphically, he describes it as if you have something causing you to drop the amount of bicarb in your system, then you're going to blow off more CO2 to get you back to this line. And you can do this sort of every different way. You can, if you have too much bicarb, then you're going to retain more CO2 to get you back to the line. And you can do this, all the way around. If you have too much, if you start off with too much acidosis pushing you this way, then the body is going to retain bicarb to get you back to the line. And so in sort of the physiologic traditional way of looking at it is just that kind of looking at that. And then and this is from another article. This isn't one that I sent you. I also had Dawn send you guys a couple of articles. One of them was talking about like strong anions as being sort of a newer mechanism of looking at acidoses. And that was like more of a published article looking at if they looked at different ways of looking at it, would they miss more or catch more of the abnormalities? And then the other one was from the ASA has refresher courses every year at the National ASA, and they publish little essays sort of summarizing each of the refresher courses, and those published essays are usually on topics that are relevant to us, obviously, because it's at the ASA, but they're also really good for reviewing for like board kind of stuff, because the types of things that people put in those refresher courses, they're doing them sort of aiming at what's important, the same things that are important to the people who are writing the boards. So the little tidbits that end up in those refresher courses happen to be really board testable kind of information. So I added one of those too. This came out of one of those, but not the one that I sent you, which is basically sort of the expectation that that compensatory mechanism that we're talking about, if you have a primary respiratory acidosis, for every 10 millimeters rise in PaCO2 above the normal 40, you expect in the chronically compensated person for their bicarb to go up by 4 milliequivalents. So anywhere up to 4 milliequivalents would be normal compensation. You're either acutely in the situation and not compensated yet, or you're gradually compensating up until about 4 milliequivalents per liter. And for respiratory alkalosis, so if you have some sort of neurologic injury that's causing you to overbreathe, then for the drop, you would expect to see the bicarb drop by about 3 milliequivalents once it's compensated, so not quite as much. And then for primary metabolic problems, so a primary metabolic acidosis, the change in CO2 that you would expect, you can calculate by this here. You can take the bicarb that you end up with after your metabolic acidosis, multiply it by 1.5 and add 8, and that should be your compensated CO2. And that should happen quite quickly. So they don't talk about really acute and chronic compensation here because your respiratory compensation should be really quick. You have really sensitive chemoreceptors in the brainstem that drive the respiratory drive, and just a change in pH of a small amount, 0.1, will change your minute volume by 50%. It'll increase it by 50% or double it for just really small changes. So generally, if you're capable of compensating respiratorily, that compensation is probably going to occur before you can draw the blood gas to look at it. So that would be the expectation for your PCO2 would be for it to drop to this level if you have a metabolic acidosis where your CO2 is low. If your bicarb is high, you can multiply it by 0.7 and add 20, and you would expect your CO2 to rise to that amount, basically. Of course, we see problems where you don't necessarily see compensation but if you have two problems causing an acidosis, both metabolic and respiratory, you can pretty quickly get in a bad situation where you can't compensate and your pH can get dangerously low, as shown there. Same for alkalosis. There's examples, someone with ARDS where you also have NG drainage. Oh, this is, sorry. This is, he was, had this slide in here saying you can also be tricked. So you can look at their pH and you can see that their pH is normal, and you might, but that doesn't necessarily mean that there isn't primary problems going on. You can have a primary metabolic acidosis with a primary metabolic alkalosis, and they can sort of cancel each other out for a pH that's normal, but your CO2 may be high and your bicarb would be high at the same time. So, just looking at pH may not help. So the physiochemical approach, which is sort of the one that was being pushed in that article that I sent, talks about strong anions being a large component of acid-base physiochemistry, and basically they suggest that many of the acids that are in the body represent unmeasured anions, and that you can calculate those and find them as anions. An anion gap, the anion gap that people talk about, is a representation of that. Depending on your source, some people don't use potassium in the calculation of this. Some people just use sodium. But you add up the sodium and subtract away the chloride and the bicarb, and you have some measure of unmeasured anions in the blood. But there's a lot more anions and cations in the blood that have to equal each other, have to balance each other out. And so, like the unmeasured cations, depending on whether you measure potassium or not, most of the time people don't measure calcium, magnesium, and those small amounts of hydrogen protons. And most of the time people don't consider the proteins, the phosphates, the hydroxyl groups, the sulfates, the carbonic acids, and all these other inorganic, non-volatile acids that act as anions in the system. And they're sort of represented here, that you have all your cations over here have to equal all your anions, and you have unmeasured cations over here, and you have unmeasured anions over here. And if you and there's the unmeasured cations are supposed to match up here, and the amount of unmeasured anions that are not balanced by the unmeasured cations ends up being that anion gap. And so the anion gap usually includes proteins and or sort of approximates the amount of these other anions, the proteins, the phosphates, and the other inorganic acids in the system. And we expect that to normally be about 12 plus or minus 4. And if you have an increase in the amount of these inorganic acids, then your anion gap will go up, and we say you have a anion gap acidosis versus a non-anion gap acidosis if this was normal. So that's one way to look at this. He's pointing out here that if you have increased inorganic acids, then your body's response to that is to waste more bicarb through the kidneys to sort of balance out the electrical neutrality. And you end up with less of your bicarbonate buffer, which is your major buffer, and you end up being more acidotic and as a result. So you'd have an increased anion gap, and you would see that on an ABG, you would see this one in particular because it looked like an acidosis because the bicarb was low. You can also get tricked up if your proteins are low at the same time. You may not catch this because you may have an increase in your anion gap from these non-volatile acids that are sort of organic acids. But you may at the same time have a decrease in the amount of protein. So if your albumin's real low and your protein's real low, then having low proteins can sort of cancel out these increased organic acids, and you can miss that type of acidosis from these. You can miss like a lactic acidosis or ketoacidosis pretty easily if you're looking at anion gap only without considering the effect of protein if they happen to have less protein than normal. So that's what he's talking about on this slide, is your protein's decreased. So if you're using anion gap, you really should be correcting your anion gap, at least for the amount of albumin, which is something that we can easily measure. And you can, for every gram per liter change in your albumin, your anion gap should, you want to adjust it by a quarter, so to get a correction. Then there's the apparent strong anion gap, which is taking into consideration more of all these anions and cations. And we would expect that we can calculate this. This is apparent to us, so that's the strong anion difference that's apparent, and the strong anion difference that's effective also would include these extra acids that we don't normally measure. And so the difference between those is going to be those inorganic acids. But you can calculate the effective strong anion difference by this calculation here, and then you can compare that to the strong anion difference that's apparent here, and identify different causes for metabolic acidosis. And so they've listed some of these here. Generally, your anion gap is going to increase along with that strong ion gap difference at the same time, and those are the mud piles types of causes that most people remember. Does anybody remember what mud piles is comprised of? There's like lithium ions and ionized or something like that. Okay, and so he just named off all these things that are here. So that was the physiochemical approach, which is what's in the article that I sent you. And then Horst really likes to talk about base excess, and base excess is basically they normalize the plasma to a CO2 of 40, and then they figure out how much acid or base has to be added to the plasma in order to make it the pH that we expect of 7.4. And the amount of acid or base that you have to add in becomes the amount of a negative amount of base excess or a positive amount of base excess that's required. So that takes the respiratory component out and just leaves you with, do you have a metabolic acidosis or alkalosis? And the base excess is positive if they had to add acid in order to get it back to normal. That meant that there was excess base, so the base excess is positive in that situation. So that would be an alkalosis. So he has a lot of slides showing all the different etiologies and how that works on the graph. These take time looking at acid-base stuff and calculating it and figuring out acid-base. Takes a lot of time to just kind of read and think through. I don't know that it helps me to read through these slides for you any more than we already have. It's already probably stuff you've seen before. One thing I didn't see, we can go through here and see if it comes up, but one thing that was mentioned in the book, Morgan and McKayo, was about the use of bicarb in code situations being sort of discouraged, and that is true. That if you're not moving blood through the cells, and you add bicarb to the blood, the bicarb can dissociate and produce CO2, and the CO2 can readily go into the cells and cause acidosis in the cells. So unless you're removing the CO2 and keeping the circulation going and keeping the ventilation going in order to remove the CO2, adding bicarb to the situation doesn't help anything in a sort of an acute arrest situation. But once you have perfusion and you have respiration, adding bicarb does help buffer the situation, and the amount of bicarb to give can be calculated. It's basically 30% of the body weight in milliequivalents times the base excess. So if you look at the base excess on the ABG, or if you just take the difference between the normal bicarb amount, which is 24, and whatever it is, so if your bicarb is 17 on your blood gas, then you need basically 7 milliequivalents per liter, and you multiply that times the 30% times their kilogram weight to give you how much bicarb you should actually give. So that would be, say, so if it was 7 and they were 70 kilograms, that would be 490, and a third of that is like so somewhere in the 120 range, 120, 130 or something, I don't know. So you'd have to give over 100 milliequivalents, which would be each amp of bicarb, each thing that you have, I believe is 50 milliequivalents. So you'd give 2 or 3 amps of bicarb in order to correct that. We normally just sort of, in the critical care arena, let people have permissive hypercapnia, but you can also adjust your ventilation. We normally don't treat with bicarb unless they're acidotic, worse than 7.2. Some people say even 7.1, and they have to have good ventilation to blow off the CO2. We didn't talk about THAM, but THAM is sort of it's a buffer that doesn't produce CO2, and so there's no CO2 that has to be eliminated for it to buffer the acid. So THAM, which is a short for this long word of tris, and Horst always calls it tris because I guess that's what they call it over there. So if you hear tris from Horst, it's THAM here, and it's on back order like most other things that we use or like to use. But it doesn't have to. You don't have to depend on ventilation. So theoretically, that's better in like a code situation or most situations that we're involved in where you want to buffer it. But it does, in the long term, it requires renal excretion. So if they're in renal failure, you shouldn't keep giving this to someone in the ICU. Although you could still make the argument that in the short-term code situation, it might still be appropriate and better than bicarb. But we don't. Have you guys seen that used at all around here? Yeah, we used to use it a little bit more, but then it got in short supply, and I think people kind of fell out of use. And I don't even know if it's available now. I bet it's probably still relatively unavailable, but I haven't seen its use sort of bounce back since it became in short supply a little while ago. Sodium acetate is added to like our TPN solutions and stuff to balance it out. And it's lactated ringers has lactate, but help me out here. What's the Plasma Light has acetate in it, and that acetate gets converted to bicarbonate in the liver on a milliequivalent to milliequivalent conversion. And except for the sodium that goes along with the sodium acetate, it really doesn't cause any problems. Horace was talking about the amount of potassium in the body. So on the left-hand side is the amount of potassium intracellularly versus extracellularly being handled by the sodium potassium ATPase, maintaining that. And really 98% of our potassium is intracellular, and that's why whenever we have to give potassium to someone who has low potassium, we actually have to give a lot of potassium because we have to give enough for the intracellular space plus the redistribution into the intracellular space, which is many times larger than the intracellular space or intravascular space. But the main reason why he had this on here was to show that there's like this huge gradient built up that whenever you get into acidosis and you start making exchange for your hydrogen ions for your potassium ions, that you can get hyperkalemic pretty quickly. So whenever you get acidotic extracellularly and you start exchanging those hydrogen ions for in the cell for potassium ions out of the cell, you can get hyperkalemic pretty quick. And I think I read, maybe it was in Morgan and McKell or one of the other sources that I was reading here lately, that for every 0.1 change in your pH, that you can expect your potassium to change by like 0.5 to 0.6 milliequivalents per liter. So that's a relatively large change, particularly if you're already hyperkalemic. If you're already at 6 and you drop your pH by 0.2, you can be at 7 very quickly, and a change from 6 to 7 can prompt a cardiac arrest pretty quickly.
Is that like hours quickly, or?
So the arrest can happen really quickly. The exchange happens quite rapidly as well, because there's all this, like extra potassium just waiting to exchange. It's more minutes than hours. But most acidosis doesn't happen over minutes necessarily, except for respiratory acidosis. So if you're sort of on the edge and you get a respiratory acidosis quickly, that can cause shifting of potassium almost as quick as the respiratory acidosis itself. So you do have to be careful in in a renal patient who already has high potassium, letting their CO2 get too high. Let's see. So most of the time, whenever we look at blood gases, what's reported out to us by the lab is called alpha-stat, which means it's been normalized to normal temperature, 37 degrees. And most of us are used to, we don't know what the normals are for other temperatures besides 37 degrees. So whenever we look at it, we generally sort of adjust things based on what it would be if the patient was at 37 degrees, which is what we hope them to be. In the pediatric world, at least, whenever patients go on bypass, cardiopulmonary bypass, and you cool somebody down, the partial pressure of CO2 is going to drop pretty dramatically, being a gas, and that changes how what your pH measurements are, and what your normal CO2 partial pressure of CO2 is. And so there's been a controversy in the past about whether it's better to correct those people to normalize their CO2 at the temperature that they're at and normalize their pH at the temperature that they are at, like 32 degrees Celsius instead of 37 degrees, or whether it's better to use the measurements at what it would be at 37 degrees and just correct them to what it would be at 37 degrees. In the pediatric cardiothoracic literature, they basically have empirically tried to test this, and they believe they have better outcomes as far as neurologic outcomes, strokes, and things like that. If you treat pediatric patients and try to normalize their CO2 at the temperature that they're at the 32 degrees, and that's called pH stat management. Versus what we normally do in adults is normalize everything always back to 37 degrees. I don't know that it's been studied in adults. I don't think it's been studied and shown to be any different. I think it's really only been studied in pediatrics where they showed pH stat to be better. So I'm sure whenever you start doing cardiac, this will be one of the things that comes up. Generally, because we don't know any better, we treat adults with the alpha stat, and we treat peds where we do know better, potentially, with pH stat management. That was pretty quick. There's a lot more to acid-base calculations and things. I don't know that it helps. It never really helped me to hear a lot of lectures. I kind of had to read it for myself and figure it out. But do you guys have any specific questions related to acid base?
Bicarb in a COVID situation, you would like them to be ventilated and perfused.
Yeah.
Perfusion is adequate from like good chest compressions, or is this after return of spontaneous circulation that you would recommend?
It's a good question. So most people would still say that perfusion is not good enough with just chest compressions, but I think that's all relative. I think if you're doing a good job and you have a reason to be given the bicarb, it makes sense as long as you're ventilating. But certainly, where everybody's sort of in agreement is if you don't have an airway yet, you probably shouldn't be given bicarb, because then even if you are circulating it back to the lungs, you're probably not adequately getting rid of it. So. That's controversial. Most people who are in the business of codes and the quality people really don't like to see bicarb in codes. However, empirically, whenever we give it, we feel like we get results. So it's kind of hard not to give because we think we see the results. It may be that the results of giving bicarb is mainly the results of giving a hypertonic solution that kind of it supposedly is like a better bolus to the better preload to the heart whenever you give a slug of bicarb that sort of stays intravascularly a little bit better than giving most of our other medications that are just isotonic and they kind of diffuse out before they even get back to the heart. I don't know if that's true or not. I'm just relaying what other people say. So, all right. Thanks.